Good afternoon. It's a pleasure to be here this afternoon to discuss the revision to ICH Q5A, viral safety of biotechnology products derived from cell lines of human or animal origin. I'll begin with a brief outline of the presentation. So I'll start with some background on the revision. I'll then transition into key principles of the revision and then talk about the key updates that were incorporated into the document. And I'll finish with some conclusions. In terms of background, ICH Q5A was introduced in 1999, and for the past 20 or so years, it's been a very useful document. However, it was recognized that in light of scientific advances and advancing technologies, a revision was in order, and so a concept paper and business plan was introduced in November of 2019. And for the last 4 years, the document has evolved and has been changed, with the Step 4 document being released last November in Prague. So I'll go over some of the key principles of the revised document. And these include the overall retention of the basic organization and the scientific principles of the document based on the utility of the original. There was a recognition that the advances in science and technology did necessitate an update to the document. And these scientific advances that have been written into the new revision allow for flexibility for continued scientific advancement. The EWG was careful indeed to not paint themselves into a corner. The new technologies introduced into the document align with the 3R principles of replacement, reduction, and refinement of animal testing. And there's been an introduction of advances in viral safety as well. Certainly the application of new technologies such as next generation sequencing has been incorporated into the document. The introduction of platform approaches to viral clearance. New product types have been incorporated, as well as viral safety considerations for continuous manufacturing. So the objective of the document remains consistent with the key scientific and regulatory considerations with respect to virus clearance, characterization, and testing. So the three principal complementary approaches to virus safety remain intact, which are the selection and testing of cell lines and raw materials for virus contamination, assessing virus clearance capacity of the process unit operations, and also testing for infectious viruses within the product at the appropriate stages of manufacture where they're most likely to be detected. Q5A R2 is designed to be used in conjunction with other ICH guidances, namely ICH Q2, Q5D, and ICH Q13. Public consultation took place last year, and a number of the comments were incorporated into the document. A lot of them concern the new products, so clarifications regarding the new products. Some new definitions were required to take into account the integration of new concepts into the document. Clarification in terms of some principles such as in vitro cell age and end of production cells, which is a terminology that was introduced with the new products. Also implementation of next generation sequencing and some more clarity in terms of how that is being integrated into the document. Additionally, there was a removal of Annex 1, and this annex dealt with the production of product in vivo, namely ascites fluid, and this was removed. Pardon me. In terms of the table of contents, the introduction was subject to some significant changes with the introduction of new product types and new concepts of viral clearance and viral safety. Sources of viral contamination was only subject to minor changes. Sections 3 through 6, major changes took place with these sections in terms of cell line qualification, testing for viruses, viral clearance studies, and evaluation of viral clearance. A new section on continuous manufacturing was incorporated, And as I mentioned, the glossary was changed significantly to reflect some of the new concepts and modalities introduced into the document. In terms of the annexes, the first, with the exception of Annex 1 from the original document, which was removed, the first 4 annexes in the new document remain somewhat unchanged. 2 new annexes have been introduced. One detailing examples of prior knowledge with respect to virus inactivation and clearance, and a new annex detailing aspects of genetically engineered viral vectors and the testing and viral clearance that is required for those new product types. In terms of the new product types, key update 1, the scope was expanded to include viral vectors. And these viral vectors are vectors that are amenable to viral clearance. So for example, non-enveloped viruses, which can withstand some of the virus inactivation conditions. Genetically engineered viral vectors and viral vector derived products. So these are products that are where viruses are used in their production, and they are not actually the products themselves, the viral vectors. So these can be used to produce recombinant proteins, such as in the baculoviral system. Additionally, viral vectors, where viral helper virus may not be required for their production, have been incorporated. So, for example, the triple transfection of cell lines for production of AAV is a good example. In terms of viral vector-derived products, an example of this could be proteins manufactured from viral vectors for generation of virus-like particles, and also the protein component for nanoparticle-based vaccines with the protein product as the delivered product. So the overall sections were altered to incorporate this new information for new product types. For example, there was additional reference in Section 2 for sources of potential virus contamination being updated to reflect for these new product types there will be virus seed banks. Additionally, in Section 5, which details the rationale and action plan for virus clearance, a new case F was introduced regarding the clearance of production or helper virus. So in terms of the use of the relevant model virus for clearance. A good example, so the examples of viruses utilized in clearance studies were updated to include baculovirus, for example, as one such virus type that could be used for the production virus. And in terms of Annex 6 and new product types, this is a new annex detailing specifically virus safety considerations for these new product types, including information on testing in a new table where these expectations for testing for these products are detailed. Key update 3 is continuous manufacturing. So there's an entirely new section dedicated to virus safety and continuous manufacturing. And this deals with the specific aspects of continuous manufacturing that have to be considered in terms of virus safety. And it's designed to be used in parallel with the ICH Q13 guideline on continuous manufacturing. And as I mentioned, specific aspects of continuous manufacturing and how these are dealt with respect to virus safety are outlined. So for example, with cell cultivation, the length of time that the cells are being cultivated in the continuous process has to be considered. In the event of a contamination event, how the material is treated or separated, if such an occasion arises, and the thought process behind that was integrated, as well as the integration of unit operations. So how these unit operations are thought of. Are they thought of as batch operations as a starting point for dealing with the virus clearance considerations, with the virus safety considerations? Additionally, in terms of specific considerations for unit operations, such as the chromatography, if there are the chromatography columns used in tandem, in terms of low pH and solvent, what is the residence time and the exposure time of the product to the inactivation agent, the other considerations that are specific for those inactivation steps. In terms of viral filtration, considerations of the product flow rates through the virus filters, the interruptions in terms of the filtration process. All these elements of filtration have to be considered with respect to the ability of the filter to clear the viruses effectively. In terms of new test methods, the main new technology being introduced into the document, and it impacts a large number of sections of the document, is the introduction of next generation sequencing. Excuse me. There is a new section in the cell line characterization section of the document detailing molecular methods, next generation sequencing being one of them. In terms of the application of the technology, which could be used to replace the in vivo test or supplement or replace the in vitro test, it's stated clearly in the document that head-to-head comparisons are not recommended or expected. NGS is considered a limit test, and so it must be demonstrated capable of detecting low levels of a contaminant, and as such should be appropriately validated for that use. In terms of key update 4, the NGS or the nucleic acid amplification techniques may also be used as an alternative to certain virus-specific detection methods, such as the MAP or HAP or RAP antibody production tests. And that, again, is in line with the concept of the 3 Rs. So recommendations regarding the uses of NGS or nucleic acid testing are described throughout the document. Key update 5 is resin reuse, and there's been certainly a large amount of scientific progress in the last 20 years with respect to understanding of the resins being used. For example, in the protein A columns, the affinity capture chromatography, it's understood clearly now that virus removal is not impacted or actually slightly increases for used resin. And as such, the guideline is clear in stating that the product-specific resin reuse is not an expectation. As for other resin types, the guideline remains open-ended regarding the use of prior knowledge. For example, the anion exchange chromatography or the cation exchange chromatography. And so for these resins, or perhaps it's a little less clear, but there may be some advances in this regard. Equivalent prior knowledge should be provided in place of product-specific viral clearance studies. So key update 6 deals with prior knowledge. So a new section has been added in section 6 containing specific principles required for the use of prior knowledge. So, for example, a well-characterized platform process must be used. So, speaking to a large amount of experience with the particular process. The composition of process and intermediates. What are the impurity levels? They should be similar with the similar process. In addition, the order of unit operations should be the same, with an equivalence of the upstream step to the one being validated for viral clearance, and the robustness of the critical parameters should be demonstrated as well. In line with this, a new annex has been created, Annex 5, that outlines 3 virus inactivation or clearance steps and the process parameters that have been characterized in terms of their effectiveness for virus clearance or inactivation. And they are regarding the low pH detergent solvent inactivation and also the virus clearance with the virus filter. Key update number 7, the flexible approach for well-characterized substrates, details testing flexibilities for cell substrates that have a lot of prior knowledge, such as CHO, for example, which has a long history of safe use, and is also the retroviral particles have been demonstrated to be non-infectious. Annex 4 includes a footnote in the safety factor calculation where the safety margin is indicated that a margin of less than 10 to the minus 4 particles per dose may be acceptable for these particular cell lines. Additionally, the use of CHO-derived endogenous virus particles may be used in virus clearance studies, with the caveat that the molecular or biochemical detection method be qualified and demonstrated to be fit for purpose. In addition, in vivo testing may be excluded in certain situations. For example, where a parental cell line has been used to manufacture or produce a number of master cell banks, and also the controls for the generation of master cell banks from that parental cell line are well characterized. So it's clearly stated in the document that in vivo testing is not necessary for extensively used, well-characterized cell lines such as CHO, NSO, sp20, based on prior knowledge. And the glossary has been changed, of course, to reflect new definitions for new product types and new concepts that we just spoke of. For example, next generation sequencing, and definitions regarding expectations for new products, such as the helper virus, viral vector for protein expression, viral vector derived products, and master virus seed and working virus seed and production virus. All concepts that need to be clearly defined. Additionally, the new concept of prior knowledge had to be appropriately defined, including the definitions of platform validation and platform manufacturing, process robustness of virus clearance, what is prior knowledge exactly. Additionally, as I touched on earlier, definitions to align terminology that has been introduced due to new product types such as end of production cells, and the legacy definition of in place for the limit of in vitro cell age. Those had to be aligned, as well as jurisdictional differences in terms of defining things slightly differently, such as extended cell bank. And these had to be harmonized in the document. In terms of implementation, it was stated in the document that products not specifically indicated in the scope, the concepts of the document can still be applied and be useful. In terms of validation of new technology such as next generation sequencing, the guideline has not been prescriptive in how to validate, and it's encouraged that the users of the document read the revision in concert with ICH Q2 and also ICH Q5D and ICH Q13. So in conclusion, the ICH Q5A revision establishes harmonized scientific and technical requirements for products derived from characterized cell lines of human or animal origin. And it helps outline regulatory expectations for testing, evaluation of virus safety. The guideline has retained the original structure and principles of the original guideline and provides additional recommendations on the 3 established approaches to control viral contamination, including the selection and testing of cell lines and raw materials, the assessment of virus clearance capacity of the processes and unit operations, and of course the testing of the product at the appropriate stage of manufacture where it's most likely to be detected. So with that, I'd like to thank you for your attention, and I'd be happy to take any questions.